So now I'm going to explain about the non-cyclic photophosphorylation. So the non-cyclic photophosphorylation involves of photosystem 2 and 1, which consists of antenna complex and reaction center, which is P680. So the activation begins when light is absorbed by the pigment molecule, or we can call it as chlorophyll. So the energy from the pigment will transfer it from one and another until it reach at the reaction center. So the P680 is the reaction center. After it reach at the reaction center, it will then move. It will then excite and move to the higher level until it reach the primary acceptor. So the energy excite will oxidize the P680 and become P680 plus, because it loses the electron. So the electron transport chain will consist of electron acceptor in a series which it connect the PS1 and PS2 and PS1 here. So okay, so now I'm going to explain the activation of photosystem 1. The same thing happen like photosystem 2, which is the activation begin when light is absorbed by the pigment molecule, and the energy will transfer from one to another until it reach at the reaction center, which is the P700. And then the electron in this molecule will excite and move to the higher level until it is accepted by the primary acceptor. So the energy excite will oxidize the P700 and become P700 plus. So only single electrons will pass down the electron transport chain, but 2 electrons are needed to reduce one molecule of NADP plus. So NADP plus accept 2 electrons and 1 proton of H plus, and reduce to NADPH here. Okay. The electron transferred to NAD plus is by the ferredoxin, the FD ferredoxin. It catalyzed by enzyme ferredoxin NADP plus reductase. So the NADP plus accept 2 electrons and 1 proton and reduce the NADPH. So the NADP plus as terminal electron acceptor. And lastly, the NADPH is released to the stroma. Okay, so now I'm going to explain about the Calvin cycle. So there are 3 phases, which are the first phase is carbon fixation, the second is reduction, and the third one is the regeneration of the carbon dioxide acceptor, RUBP. So the 3 molecules of carbon dioxide will enter the first phase. The carbon dioxide will incorporated 3 molecules of RUBP, which is 5 carbon sugar, 1 2 3 4 5 carbon sugars. So the reaction will produce short-lived intermediate or unstable intermediate that is broken apart into 2 PGA. So it will produce 6 molecules of PGA. It will phosphorylated by 6 molecule of ATP. Then ATP will release one of its phosphate and attach to the PGA molecule, forming 1,3 biphosphoglycerate, and it reduced by NADPH. So the reaction requires 6 NADPH, and NADPH will become NADP plus. So the 1,3 biphosphoglycerate remove its phosphate, become 6 molecules of G3P from 6 molecule G3P to 2 molecule 2 molecule G3P will be used to synthesize sugar, which is produced which is produce it will produce glucose. It will produce the glucose. So the remaining G3P will enter the third phase, which is the generation of the carbon dioxide receptor, REBP. The remaining G3P enter the third phase, and the molecule of G3P will undergo rearrangement to produce the RBP, and will be photophosphorylated by ATP. It phosphorylated by ATP. 3 mole are required. Then reaction will produce 3 mole RUBP, the molecule of RUBP. So now the RUBP regenerated and is ready to initiate the next cycle. Okay, now I'm going to explain about the CAM pathway. So when night, the stomata opens, and during daytime the stomata will close. So when the carbon dioxide enter, it will incorporate with PEP, 3 carbon compound, and then it will produce 4 carbon compound, which is oxaloacetate, and the reaction is catalyzed by the PEP carboxylase. So the oxaloacetate converted into malate into another 4 carbon compound. So the malate will then enter the vacuole and store in the vacuole as malic acid. So during the daytime, stomata close, so no carbon dioxide can enter to the cell. So malic acid will convert into the malate, and it will split to 2, which is one is carbon dioxide and one is pyruvate. For the carbon dioxide, it will enter to the Calvin cycle, which the Calvin cycle will produce sugar, and then the carbon cycle will use the enzyme Rubisco.